Jai Hind students. So welcome to the class. Today we are going to start with the topic the DNA. In this particular lecture we'll see that what is DNA actually, what is its composition and how it is made. Alright, so let's start with the DNA. If you will see about the DNA, that what is DNA actually, then you will come to know that DNA is actually a genetic material. Genetic material means a thing which transmits or transfers the character from one generation to the other. That means from parents to the offspring. So that is the DNA. Now DNA is made up of what? DNA is made up of a long polymer of nucleotides, which is also called as base pair. This nucleotide is equal to, means the nucleotide is made up of nitrogenous base, pentose sugar and phosphate group. Further in this lecture we'll see that what are these groups and what are their functions. So now you know that DNA is made up of a long polymer of a nucleotide, and this nucleotide is equal to base pair. This is also called as a base pair, which is made up of a nitrogenous base, pentose sugar and phosphate group. Now the DNA which you see, you can also call it as a polymer of deoxyribonucleotide. In this, the word nucleotide, now you know that what is this actually? The word deoxyribo, if you will add in this ribose, you will come to know this is a particular type of sugar which is present only in DNA. If here would have been only ribose, then ribose kind of a sugar is only present in RNA, but here it is mentioned deoxyribo plus Se, ribose. That means it is a type of a sugar. This is pentose sugar's variety or a type which is present only in DNA. So that is why it is deoxyribonucleotide long polymer chain. Okay. So if you have to find out that if you have got a DNA sample and you are not aware that which organism's DNA sample is it. So what you can do is, due to the modern genetics techniques, you can find out that what is the number of base pair in that particular DNA sample, and according to the number of base pairs you can find out that which organism it is actually. For example, here we are having two different organisms base pair which they are having. Okay, one is Escherichia coli. In Escherichia coli, one length of DNA is 4.6 into 10 to the power 6 BP. This is base pair. This much is the Escherichia coli DNA's length, one length. In human or the Homo sapiens, it is 3.3 into 10 to the power 9 BP in the haploid cell. Don't forget this. Okay, so this is about the nucleotide and DNA's names extraction, that what it is actually. So now let us start with the nucleotide, that what it is. So now let's see that what is our nucleotide made up of all. As we have discussed that nucleotide is made up of nitrogenous base, pentose sugar and phosphate group. So let us start with the nitrogenous base. So the nitrogenous base, which is over here, it is made up of organic molecules with some similar physical properties. For example, this nitrogenous base is made up of two things. So one of it is called purines and the other one is called pyrimidines. So we are having two different types of the nitrogenous base, purines and pyrimidines. Purine is actually adenine and guanine, whereas pyrimidine is your thymine, cytosine and uracil. Fine. So now let us see about them. The purine and pyrimidine, they make the base pairs which is called as the nitrogenous base, right? The adenine, guanine, thymine and cytosine. These four you will see in the DNA. In DNA, if you will see, you can observe A, T, G and C. These are somewhat like this in a base pair found. But here you can see uracil too. So when you will see RNA, in RNA there will be A, U and G with C. So in RNA, thymine is replaced by the uracil. Okay, so this is the difference between DNA and RNA's base pair. Now coming to the pentose sugar. So pentose sugar, as we have already discussed, that it is of two types, ribose and deoxyribose. So the pentose, one is ribose and the other one is deoxyribose. These are the two types of pentose sugar which you can see, and ribose is present in RNA whereas deoxyribose is in the DNA. So this kind of a structure, this sugar you will find in the DNA. So as you can say in this, that DNA is made up of phosphorus and sugar. So its base pair, its skeleton, its body is made up of because of the sugar and phosphorus. Now let's see the third part of which DNA is made up of. DNA phosphate group is made up of, phosphate group means the bond, the lining which is over here on which this nitrogenous base and pentose sugar are lined. That thread like structure is made up of nitrogenous base, this one, pentose sugar which is linked through the N-glycosidic linkage. Alright, so N-glycosidic linkage adds nitrogenous base and pentose sugar to form this phosphate group. For example, adenine plus ribose sugar is equal to adenosine. Adenine plus deoxyribose, that one, is equal to deoxyadenosine. So now here you have come to know that what is the structure of DNA. Alright, so now you can say that a nucleotide which is formed, so the nucleotide because of which linkings DNA is made up of, so you can say that a nucleotide is formed depending on the type of sugar, right? Maybe deoxyribose or maybe it can be the ribose. So depending on the type of a sugar, when a phosphate group is linked with the 5-OH of nucleoside, so maybe this over here, alright, so it may be on the 5-OH group of the nucleoside through a phosphoester linkage through this one, right? So you can say this now, and this is to form the dinucleotide. In the same way, nucleotides may join to form a polynucleotide chain too. Polynucleotide chain means same like this only. If many things are joined together, they are going to make polynucleotide chain. This is going to make a nucleotide. If so many nucleotides are joined together, they will be called as a polynucleotide chain. So this was about this much. Now further we'll see the structure of DNA. Thank you for watching the video.